the very modern way that this kind of science is done, okay? And really what you're talking about is bioinformatics. Just off the top, looking at the name, can anybody guess what bioinformatics is, or do you already know?
About studying the formation of life.
What is it? Studying the formation of life? That's a good guess.
Application of statistics and computer science to the field of molecular
Oh.
Okay, great.
There you go.
So there are a lot of definitions of bioinformatics. The National Institute of Health, the federal agency that is really in charge of a lot of the biomedical sciences that happens in this country, they have their own definition. I've kind of boiled that down for you just a little bit to think of it this way. So it's the use of computer-based tools and mathematics and statistics to study biological information. Okay, bioinformation. Okay. Another way of saying bioinformatics, by the way, is computational biology. If you're a computational biologist, you spend more time developing the computer-based apps and the statistics that we as scientists, biologists, use to study our organisms, okay? So the bio part of this can mean anything that's living, right? So from very simple organisms like bacteria and yeast to more complex things like plants and, somebody know what that is?
A turtle.
You said a turtle, not a turtle. What'd you say? Scales.
A cell.
Hair.
A cell.
Which one did
Cells.
Which one did you point to?
Or an
The
atom.
top one. The top.
An atom.
It's algae.
Bacteria? Oh my god.
It's algae.
Algae.
So more complex organisms like algae and plants, and of course also mammals, humans, other animals. Okay. Anything that's living can be analyzed and studied with the use of bioinformatics. Okay, the informatics side of it is really about taking information that we as science biologists gather from these organisms and converting that into a digital format, okay? Really converting that into something that is computer-based. And then using very powerful computers to analyze it. And I'll show you more of what all of this means in a second. But before I do, let's think back about, let's go back through the history of science, really. There's some key steps along the process that really made this possible today. So bioinformatics really is all about being able to compare organisms. Compare two humans, or compare two snakes, or to compare a human to a snake, or to compare a human to a snake to a bacteria, or a human to a snake to a bacteria and a virus. It's all about comparison, and it's usually done at the gene or protein level. So comparisons in science is nothing new. Who's this guy? Does anybody know who this guy is?
No.
Santa Claus.
Yes, Santa Claus.
Nobody knows who this is?
Santa Claus.
Yes,
It's not Santa
scientist.
Claus.
Music.
It's not Newton. It's
Darwin.
Darwin. You heard of Darwin?
You said it.
I've only seen younger pictures.
The younger pictures, okay. In the time of Darwin, there were comparisons going on too, right? But at that time, animals were being compared and plants were being compared basically on the basis of morphology, what they look like. Also compared their habitat and things like that, right? But it was a really macro way of doing it, right? The large scale way of doing it, making comparisons. Along came this lady, Rosalind Franklin. Anybody know who that is?
I've heard of her.
Yeah.
You've heard of her?
Yeah.
You should know her. Rosalind Franklin. Okay, what about these two old farts up here? If I tell you who these two guys are, maybe you can tell me who Rosalind Franklin is. That's right. It's Watson and Crick. Also,
She helped us with the little prize and stuff.
What is that?
the Nobel Prize? No. Well, she did the work that allowed them to win the Nobel Prize. That's correct. So what was the work that she did?
Did she have anything to do with DNA?
She had something to do with
X-ray.
it.
She x-rayed DNA.
Exactly.
And died of radiation.
So this is actually a picture from her notebook. All right, so this is back, late 1800s, early 1900s. This is around the time of Mr. Taylor and I's parents, back when they were born. So we're talking about the mid-50s, when the structure of DNA was first elucidated. All right, what that means is discovered and proven. using these X-ray graphs. And then around the time of Mr. Taylor and I being born, this guy came on the scene. If you know who this is, I'll give you $1.
Ooh.
Mr. Taylor's
Yeah,
dad.
this worth taking a shot.
No, it's not. That would be a weird name.
Okay, this guy's name is Kerry Mullis. Okay, and in the early '80s, Dr. Mullis actually is the one that developed a technique called PCR, polymerase chain reaction. And essentially what it does is allow us to take just a very small piece of DNA from an organism and multiply it by billions and billions of times. So we've got billions of copies of this same piece of DNA so that we can analyze it. And that's really the innovation that allowed for all modern sort of molecular biology, okay, including bioinformatics. All right, so try to remember this guy. What's his name?
Kerry Mullis.
That's right, Kerry Mullis. I'll ask you that in a few, probably weeks, and see if you guys know.
Can you give us $1?
Maybe. If you can remember in a couple weeks, yeah.
I got you.
Okay, so that's kind of the history that led us here, and then you got to also remember that modern biology really encompasses a huge number of fields, many fields. Everything from studying the whole organism, the whole animal or plant or bacteria, to studying individual cells, and even the way that cells communicate with each other through these signaling networks, okay? Obviously, DNA and RNA and proteins are important, and then the environment that these animals live in. Also, there, in most cases, are some biomedical implication, okay? Some either treatment of disease or being able to use these animals to study some disease or some metabolic condition and so forth, all right? And those sciences are often done by individuals who don't communicate with each other, okay? So you've got people that do nothing but biochemistry, or you got people that do nothing but develop drugs and pharmaceuticals, or you've got people that do nothing but work with animals, or do nothing but study behavior. Then you've got this other set of people, scientists, who are computer scientists and mathematicians and statisticians, that are, for the longest time, have been sort of off on this island. There was not a lot of communication once you develop your science between each other, until bioinformatics. Bioinformatics is really this bridge that brings together all these disciplines so that an animal scientist and a person that develops drugs can work on the same project, okay? They can get the same government funding to do a project. When they publish their results, it's applicable in both of their arenas, okay? So what are some of the applications of bioinformatics? There are many. There are literally dozens and dozens of things you can do with bioinformatics. For the purposes of this class, we'll really focus on just a handful. The first thing, and probably the most common thing, is to study the sequence of DNA itself, okay? So we're talking about being able to take those nucleotides. Nucleotides are bases that make up DNA, right? So what are the 4 nucleotide bases that we're talking about?
Guanine, cytosine,
Guanine,
and adenine, right? ATCs and Gs, all right? And taking those ATCs and Gs and translating those into the amino acids that they code for. Because remember, as a part of translation, the ribosomes take their DNA, actually at that point it's mRNA, and then they build proteins, right? Those proteins are made up of a series of amino acids. That process at one time was done by hand, and you're going to do it by hand, and you'll see how difficult it is. So for instance, this short sequence of DNA can be translated into this short sequence of amino acids, okay? Each one of these is an indication of a different amino acid, or is an abbreviation. A second application is to be able to compare genes from different organisms. Like I said, you can compare a human to a snake to a plant, or a butterfly to a polar bear, whatever, okay? And I'll show you a little bit how to do that. So what you do is you take the gene from the human, let's say. You take the same gene, let's say this is a piece of cytochrome C. I know you guys talk about cytochrome. Of course, cytochrome C is like 40,000 bases long, but say this is a piece of it. You take it from human, and then you take the same gene from shark, and then you put them on top of each other. You line them up, okay? What do you think these stars and dashes represent?
Stars are the same.
Exactly. This is what you've already got it then. This is what is known as a multiple sequence alignment, MSA. And this is hugely important in bioinformatics. Why? Because it tells us which part of these genes are highly conserved throughout evolution. So in this example, the first 4 bases here, the A, T, G, and A, are conserved throughout evolution in both the lineage of the human and the shark. You understand?
What do you mean by highly conserved in evolution?
What I mean is that, okay, thanks. What I mean is that they had a common ancestor that had the same sequence in its cytochrome C gene. Okay?
So little change over time between them.
Yep. And that's evidence in my mind of evolution. So this is vitally important. And this is the kind of thing that will allow us to understand better these genes, not only in the human, not only in the shark, but in all animals. Okay? Yep.
Do you have a dash where you should be a star?
Oh, do I? Great. Where? Here? Here. You're right. You're absolutely right.
Cheers, Darwin.
Wow, you're getting all the stickers. Okay. Okay, I'll change that. Thanks. Okay, so also you can use bioinformatics, and you'll see this to understand what the function of the gene is. Suppose we get DNA and we don't know what it does. We don't know what the gene is. You can use bioinformatics to give you some clues on what that does. Particularly, in most cases, the full gene has a function, but individual parts of the gene have functions also. And so in this example, this may be a part of the gene or a part of the protein that allows this to bind to glucose, for instance. That's just an example. Okay? And then bioinformatics can be used to develop those phylogenetic trees. Remember the phylogenetic tree I showed you guys? On last week, the circle, that was developed through the use of bioinformatics. That people didn't just sit down and guess which animals are closely related to each other. It's all based on mostly their DNA structures, okay? And typically mitochondrial DNA. Anybody know why mitochondrial DNA is so important, or what's unique about mitochondrial DNA? Mitochondrial DNA is the DNA that's in the mitochondria of a cell. And you only get that DNA from your mother. Okay? So everybody's mitochondrial DNA in here came exclusively from their mother. Their father had nothing to do with it. Anyway, so it'll help you to build these phylogenetic trees. Okay? So let's try it. That's just a brief introduction. Let's actually try it and use it. You've got your computers open so we can do this thing. Okay, so suppose that we are a class of biologists in which I am the lead biologist. I'm the lead PI, the principal investigator. And we want to study, we're marine biologists, and we go down and study animals in the South Pacific, okay? And then we come across, as a class, we come across some new animal called since we discovered it, I get to name it, and we're going to call it the Tyusfish, okay? By the way, that's not the Tyusfish, that's the beluga whale. And this is a picture from the Georgia Aquarium. That's not the Tyusfish, this is the Tyusfish. Okay, so say we discover this Tyusfish and we want to get more information about it. We've already done the kind of morphological things, right? We've already, we know exactly its color, we know its environment, we know its size, we know how many bones it has and all of that stuff, okay? But we need to dive a little bit deeper into the DNA and be able to compare this animal to other animals, okay? To get some understanding on where this fish fits into the whole evolutionary path, right? So how do we do that? We take DNA. Where are we going to get DNA from? What kind of cells are we going to get DNA from? Yes, anybody? Blood? Anywhere else? Saliva? What's that? Brain. It doesn't matter. Because DNA is the exact same DNA is in every cell. There's one exception, and if you can tell me the exception, I'll give you a Snickers.
The tail.
The tail. There's one kind of cell that doesn't have the full exact copy of DNA like every other cell.
When you're talking about
Only one in the whole body?
the
Only
reproductive
one
cell.
kind of cell. Huh?
The reproductive cell.
That's right. That's right. Watch your head, man.
Thank you.
Okay, so
Reproductive.
in this case, it's a sperm cell because we know that that's a male. Right, but every other cell works just the same. Now, it's
really,
easier to
other
get
female.
DNA from blood, for instance, than it is from liver, but it can be done. Okay, so let's pretend like we got DNA. Can you get the light for me?
Yeah.
Okay, so we've gone to the laboratory, we've taken blood and tissue, and now we have analyzed it. That DNA, we've used PCR to amplify it and make millions and millions of copies. We've used a sequencer to actually read it, and now you guys tell me what the sequence is. So start here, we'll go down the rows. Give me either an A, a T, a C, or a G.
A.
Okay, next. T. A.
C.
C.
G.
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 A.
A.
C.
C.
T.
T.
A.
A.
G.
G.
T.
What is it?
T.
T.
C.
Stop.
She did too.
Z.
C.
C.
T.
T. C. C.
A.
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 G.
G.
C.
C.
T.
T.
C.
C. Another C.
T.
T. A. Okay. We'll give it now. Okay, that's our sequence. That's our DNA. Everybody's going to test this. Now, there's something that's called an open reading frame. When we take this DNA, we know that this DNA can be translated into a protein sequence, an amino acid sequence. Does anybody remember how to do that? Open up a Word file and everybody type this out exactly as it is.
Are you want to type
Microsoft Word.
You know what's happening on Twitter?
I don't care. Yeah, it can be Twitter, whatever. Just anywhere that you can type. That's fine. Just make sure you get the sequence exactly, okay? Has anybody ever heard of a codon or a triplet?
Yes.
Okay, good. So a codon or a triplet, aka triplet, is just 3 of these bases, right? So if we started at the beginning, it would be ATA. And the next codon would be CGG. And the next one would be ACT, and so forth, right? Those triplets can be converted, the 3 of them, into 1 amino acid. And we do that, everybody has it typed in?
Yes. I sent it.
We do that with the use of a,
How cut me off? Yeah.
okay, we've gotten DNA, we've extracted the DNA, we've analyzed it through PCR, and we've sequenced it. By the way, this is what the sequence comes out, this is what it looks like when it comes out, a series of these peaks, and those peaks give us the actual sequence of DNA. And we've just done that, so we all have our sequence of DNA, A, T, C's, and G's. Okay, this is what you're going to use. So I want you guys to translate this amino acid sequence, this DNA sequence into amino acid sequence using this chart. This is how it's done. You take the first letter and A, you find A. Here it is right here. Okay?
And this, like, you go on throughout the whole thing?
No, we see the.
Like you find the A in the first base, and the second base you find the next one, and then the third base you find the last one.
Exactly.
What?
So the first one was ATA, right? So you find A. Of course, this is RNA, and in RNA, thiamine is replaced by uracil, right? So everywhere that there's a T, you use a U, okay? So you find the A, you find the T. So we're talking about this block right here, right? And then you find the third letter, which is also an A. And if you look down here, this is the line right there. Okay? So ATA is isoleucine, the letter I. Everybody follow that?
So we put I.
So you'll put an I. Now this first row, in fact, this first two rows, you'll do it exactly like that. You'll start with the very first three, put the I. Do the next three, put whatever letter that is. The next three, and so forth. These two rows, you're going to do what they call a frame shift. Because it's possible that our DNA, our gene doesn't start with that A. It's possible that our gene actually starts with that T. The second one, the second letter in our DNA sequence. You know what I'm talking about? And if that's the case, you still have to do it by triplets. So you're going to do TAC. That'll be your first one, and then you'll do the next three, and the next three, and so forth, right? And then the last group, you guys start with the second A, or the third letter. You got me? Huh? Okay. And do the whole sequence, not just the first one. Do all of them. At one time, this is how it was done, by hand, using this chart. Using this exact same, this chart's been in existence for a long time.
Where's T?
Remember, uracil, thymine T is replaced by U when you're talking about a single-stranded molecule. You guys start with TAC.
We had one letter left over.
So what we doing?
Okay, that's fine. Yeah, just leave it.
Can you explain again?
I'm confused.
I didn't hear you.
Like what we doing with it?
Okay, so you are on the road that you're going to start with T. Did you write it down? You write the sequence down? So you're going to start with here, TAC. Now using that chart up there, find TAC. I'll show you. Just write the word stop. This is the first base, so this is T. Alright? T is here. And then you find A, second base, so TA, and then you find C. And C is the second row.
Oh.
So your first one is going to be tyrosine, or the letter Y.
Oh, so you'll write down?
So you write that Y, and then you'll go to the next three and do the same thing. So that you should get a amino acid sequence of at least, or I guess about 8 bases, right? Is that what you guys got?
So be T. IRT. Just IRT. So it's IRT.
Do the whole sequence. Do the whole sequence, right. Make sure you do the whole sequence, not just the first base, not just first amino acid. Yeah. Type the word stop and then continue going, okay? Did you guys get any stop codons? Right, so you haven't even heard of a stop. Did you get any TAAs or TAGs or TGAs? Good. T is you. Right. C is C. Yeah. C is C.
Stop and just start with the ones after that.
Johnson?
You got a stop codon? Oh, you got the whole sequence?
I'll fill her.
We're out so long.
She'll be no good.
I'm sorry. Okay.
CAA.
G is G. All the letters are the same except for T is replaced with U.
So it's Q. It's Q.
Okay.
I'm sorry, I'm trying to do this technical.
Tell me which ones gave you Q.
Okay. The first 3 letters, ATA.
Tell me which ones gave you
A
K.
on the right side,
AH.
break down for A on the right, on the top. And that puts you where asparagine is. See that? So find the last letter, which is A. So what? A.
See, he has AG.
Oh, can I get it?
You
So,
have GC.
first we look up the red one and then the blue.
I'm sorry, you're looking at ATA. Yeah, look at the red and the blue and the middle. So you're ATA, so you're here, here, and then here. So you put I.
That's probably a good way to do it. So Kevin did something good by spacing the codons.
That's what Edwin did too.
It makes it real easy to see which ones you need, okay? Our bell is about to ring, right? Is the bell about to ring?
We got 6 minutes, yeah. No, just leave them out actually.
Let me,
You gonna fail one, right?
oh,
Yeah.
that's fine, but what you wanna do is
No.
have a sequence at the end that is.
We're just gonna leave the computers where they are,
Right?
What's the title?
and I'm gonna actually have the students come plug them in.
So you gonna explain what the assignment you gonna do?
Oh, yeah.
So you wanna end up having a full sequence like that, okay?
I'm busy next week.
Because you're gonna use that to actually compare. If you're successful and have done
I
this,
need my graduate.
that's great. Save it, and we're gonna open up a new web page and type, and you can Google NCBI.
And we're gonna do 2 pages. This show this morning.
Can I write at the corner?
We didn't show
Right
up, but earlier this morning we gotta do 2
here? Even though
pages.
it's on the smart.
Yeah, it doesn't matter.
If you're done, go ahead and Google NCBI Blast. This is an app that will allow us to copy and paste our protein sequence, our amino acid sequence, and then search a database to find out what gene this is. Okay? Blast is called the Basic Local Alignment Search Tool, and it's made by the National Center for Biological Information, NCBI. So if you go to Google and type in NCBI Blast, it should be the first thing that comes up. Search tool, yep. So go ahead and go to that. NCBI. After that, copy your amino acid sequence. Oh, I'm sorry. Scroll down to the bottom of the page on Blast and find Blast P. Or actually, they didn't say Blast P anymore. Protein Blast. No. Once you go into keep that freak out top of the page. That's it. So go into that. And at the bottom, toward the bottom of the page, you see protein. Yep. So, do you guys all have that? Yeah, and then you're going to copy and paste your amino acid sequence that you have translated into that box that says query. Go to Protein Blast. And then copy and paste it into where it says enter accession number or fast thing.
Yeah, sure.
Yeah, go into that.
I have a question. After that is
Go to the bottom of the page and hit search. All right.
Okay, I was like, please and walk like closed out.
Where did you say please?
What is, I'm sorry, what's it called? Blast. I'm sorry, Blast. Great. So, okay.
If we don't finish
We'll finish it next. Yeah, you can do it at home, but we'll finish next time too. Just save it. Just do you guys get the same computer every time?
No.
Okay, great. It's actually doing the search. Okay, great. That's the gene that you need. These are like I'll talk about this next time. But great. So scroll down. This is the list of the genes that you need. Yep.
Is it ready?
Yep.
I don't understand. It's just against the law.
Leave your computers on, do not turn them off. Do not turn off your computers. And just shut it open. Push the button. Turn it on.
Shut it down.
Turn it down.
Okay, so next time we'll finish this.
Yeah, this is cool.
You'll get an idea of what our gene is. We may have to create a new sequence unless you guys can write it down on paper and remember it or whatever. But I think everybody should say bye to everyone.
Bye.
Edwin, don't do that, you're gonna start crying.
Don't cry.
Yeah, I see it up here.
Don't cry.
Wait.